Hello, this is Professor Christian Holzer again. So today we are going to talk about Tirzepatide, which is on the market under the name Zepbound and Mounjaro. So what is that and what does it actually do? So Tirzepatide is a new drug on the market for diabetes. It's marketed under the name Mounjaro, but it's also on the market as a weight loss drug. It's called Zepbound. And this is slightly different from the previous drugs we talked about because this is a double agonist of GLP-1 and another peptide called GIP receptors. And we know in the clinic that this dual agonist is more effective than Semagutide or the other old school GLP-1 receptor agonists. So I hear you asking, so what is GIP? I've already told you about GLP-1, and GIP is like the sister hormone of GLP-1. They're both members of the same family. And both of these peptide hormones are released in the gut after a meal. And the reason is that they're both signaling the availability of energy, sugars and lipids. And so they really work as a team together. So it makes sense to bring them together in one drug. So what's the difference then? And I've told you already, GLP-1 is more focused on controlling blood glucose levels, and it enhances insulin release in the pancreas at high blood sugar levels. Then GIP appears to be less focused and active on the blood glucose, but more on the lipids, the fats that we take up with the food. For example, it controls the uptake of lipids and the storage in the fat cells, the adipose cells. So it's clearly related to metabolism and energy storage, but in a different way. Now, one issue is that GIP signaling seems to be desensitized in some people with diabetes. That's why they haven't really used that to treat diabetes in the past. But then they found that GLP-1 agonists can normalize, resensitize the GIP signaling. So then they thought, well, maybe we should try some dual GLP-1 GIP agonists. Maybe they're better. And there's an interesting effect that's important here. The receptors are often expressed on the same types of cells, and then they can work together. If you activate both of them, they start to form dimers. So they really work as a team, and then this boosts the cell signaling on the inside of the cell. It's like a turbocharger, and the effect is much enhanced. So in the brain you find those receptors as well, and both peptides are expressed in the brain. And on neurons, the receptors are both expressed too. And I have shown in previous studies that GIP receptor agonists, they're also neuroprotective, just like GLP-1 receptor agonists. So then, the question is, how about the dual GLP-1 GIP receptor agonists? Do they add something to the picture? Are they more effective? And here's an overview what happens in the neuron. It's a very busy picture. Don't let that confuse you. I'm just showing you this to tell you it's complicated. Like most things in life, it's not as simple as I'm making it. There's a lot more to the picture, but what you can see here is that these two types of receptors, they can form a dimer, and then they can activate a lot of signaling pathways that then control things like energy utilization, cell repair, synaptic activity, and all of these important things that protects the neurons. So here's an example where we tested different types of drugs. So we tested liraglutide and then 3 types of dual agonists, DAGC1, DAGC4 and DACH5. So they're all slightly different. And you can see that in protecting these neurons, these are dopaminergic neurons in the brain, and they tend to die in Parkinson's disease. This is what we think causes Parkinson's disease. And you can see that liraglutide is not as good as these dual agonists in protecting the brain. So there seems to be a distinctive advantage. So this is what I just said, but I'm just presenting it as a slide that you can read it in your own time. And we also looked at Alzheimer's disease, like how about protecting the brain in Alzheimer's disease? Are the dual agonists better? And we ran a number of studies. Here's just an example of a memory formation, learning a complex spatial task. And sure enough, liraglutide has an improvement, and we know it works in patients with Alzheimer's disease. But the dual agonist is better, so it suggests that it might be even better in treating Alzheimer's disease. We also looked at the amyloid plaque load. Beta amyloid is the main biomarker of Alzheimer's disease. And we could show that the amount of these amyloid plaques is much reduced by the dual agonist, and liraglutide only has a small effect. So in total, we can say dual agonists are really more effective than single GLP-1 receptor agonists. So we actually have a dual GLP-1 GIP receptor agonist in a clinical trial at the moment. And of course, you have to test them in patients in clinical trials under controlled conditions. There's only one way to find out. So watch this space. I will keep you updated on the next developments. And that's all for today. Thank you for watching, and you can always find more information on my website or actual research papers. If you're a scientist, have a look at my ResearchGate website. You can download reviews or papers on specific research topics for free. And I hope to talk to you very soon.